Hello, thank you very much for inviting me. I look forward to presenting some of the work we're doing on basically human-based models for preclinical testing. All right, so I'm going to talk mostly about the work that we've done at Hespros. It's a company founded by Mike Schuller and I. About 8 years ago, we licensed a whole bunch of patents from my academic lab at UCF and his lab at Cornell. We've used a lot of SBIR grants to push the technology out to the point now where we're working with many pharma companies, national, international. We got a 14,000 square foot facility in Orlando, Florida. Got about 50 people at the company at the moment. We are CRO. Our motto is basically we can custom build what you want based upon your specifications. And lastly, we're the first company to take a fully human on a chip model and get a drug repurposed into a phase 2 clinical trial with Sanofi. Okay, that was done about 2 years ago, and that was published last year. And so what do we do that's different is we work on this idea of clinically relevant functional readouts. You go into a doctor's office, he doesn't immediately stick a needle in your arm to be able to pull out fluid, look at biomarkers, right? So it's how are you walking? How are you talking? He checks your reflexes. He listens to your heart. He asks about elimination functions and things like that. What he's asking you about is functional readouts. So what we actually try and do is we look at muscle contraction, electrical activity for cardiac cells, neuronal cells. We look at neuromuscular junction formation, which I'm going to talk a lot about today. We can also look at barrier integrity. This allows functional analysis of these systems, both acutely and chronically, without cell death. And this is a key point, is we're not basing this upon death markers. We're basing upon things you would expect to see in a clinical trial. And so basically what we're doing is neuronal cells on top of microelectrodes, patterning cardiomyocytes on top of microelectrodes, putting muscle and cardiac cells on pieces of silicon. So when they contract, they deflect the laser beams, we actually get the force. We still look at biomarkers. We can also look at tear in these systems. And so what we can do is we can actually then culture out the cells in its own individual chips. We put them into a base platform. We can then put down on top of it a gasket where we can flow a serum-free media. Everything I'm working is all in serum-free medias, okay? And then we put a clear plastic top on so we can still do imaging in the system. And that is self-contained. So I'm going to talk today start off talking about one rare disease, which is ALS, and how we've been able to culture out the neurons, the muscle, and the Schwann cells for the SOD1, TDP43, FUS, and C9 mutations. We can then put that into our system where we have neurons on one side, we grow axons through the tunnels and then innervate the muscle on the other side. So what we can do is we can stimulate the muscle and actually then look at the contractions. And then for the disease, we see actually a loss of fidelity in these systems. And in some cases, we can actually restore that with a therapeutic. Okay, so you can actually see the system here. Again, we have the neurons on one side with the electrodes. We can put them on muscle on the cantilevers. We can do motion capture here, showing the axons going through the tunnels and making very nice neuromuscular junctions. Okay, and again, getting to this whole idea of clinically relevant functional readouts, like talking to somebody, Brian Waniger, who's running ALS clinical trials here in Boston, I said, well, what are some of the tests you have them do, a patient who has ALS? They said, one of the tests we have them do is they do a task with their hands faster and faster. And what we do is we start seeing spasticity as they go faster, and they start to lose grip strength, okay? So, and we can actually reproduce that in our systems, okay? Because we can stimulate this faster and faster, right? So I can look at this, and so if I actually start see skipping here, that's the equivalent to the spasticity. If I see loss of tetanus, that's the same as losing grip strength, okay? So directly relevant clinical parameters. And it's a very busy slide, but just it shows we have 3 different mutations here for ALS. And showing that here you're getting some skipping for one SOD mutation, some maintenance of the of tetanus. You see here for the SOD1, another SOD1, you have a little bit better fidelity, but kind of loss of tetanus. For the FUS mutation, you got better fidelity, but loss of tetanus. And again, to show that this is actually happening at the neuromuscular junction, we can then test and put the electrodes on the muscle-only side and showing the muscle has complete fidelity. So we know it's operating at the neuromuscular junction. Okay, we can also again look at this whole idea of fatigue index. This equation is right out of a clinical trial, basically showing that the more you lose the ability to go into tetanus, your fatigue index goes up, and we see that for the mutations. Then we actually were testing a collection of metabolic enhancers that was put together called the Diana protocol for ALS, and being able to show we can restore function with that for fidelity, but also most importantly, you see that the fatigue index went down dramatically. Okay, after about 17 days of treatment in the system. So we can actually then see therapeutic applications with the model. Okay, we've also been able to take the and differentiate out the muscle and showing that the wild type, SOD1, wild type, and then SOD1 mutations, and then put them together. You see actually the morphology all looks fine. So we're not killing these cells with the mutations. We're seeing functional deficits, clinically relevant functional deficits. Okay, you can see here, you said with the motor neuron, you see a deficit, which will be seen, but the muscle actually has a larger deficit with this particular mutation. Okay, then and in the combination is even worse. You can see with another mutation, it's not quite as bad. You see after about another 3 days in culture, you do get some recovery, but you have a combination here of muscle and motor neuron, both contributing to the pathogenesis that we see in the system. Okay, we've been able to do the ALS mutations in Schwann cells, okay, to be able to start looking at those models. And then we did a collaboration with Apellis to look at ALS. What they wanted to do was have an inflammation model of ALS. So what we did is we added microglia on the motor neuron side, and then we added the Schwann cells and the muscle, okay, and then activated or unactivated macrophages on the other side, and added complement into the system on the muscle side to be able to induce a inflammation model. And what we can actually see with that is in the non-activated monocytes, you see that there's very little deficit with a human complement, but their molecule was able to actually increase it. There is some deficit with the complement present, but not that much. But when you activate the macrophage, you see a huge deficit, but then they're basically their model restored that, okay, for the number of neuromuscular junctions. You then look at the fatigue index, and you can see here again, the unactivated monocytes really don't do much for the fatigue index, but you do see that there is a big decrease, okay, in the fatigue index, and that data plus the fact that we also saw the same thing with neurofilament light quantification, that also went down with the administration of the molecule, that went into an IND and an orphan drug application, which then authorized. Okay, so we have the clinical relevance of these systems at the regulatory authorities. Okay, so now we're also working now with a company, Vanda Therapeutics. Sandra's back in the corner back there. She's got a poster later on. And what we're doing is a clinical trial of one for a rare mutation in CMT, CMT2S. Okay, and we can basically differentiate out the motor neurons. We took the patient cells, took them in induced pluripotent stem cells, then differentiating them out into the motor neurons and to the muscle. Okay, and basically showing here that we have the nice markers. You can't see it over here. I guess you can kind of see it over here for MAP2 and chat. You see the nice, they turned into viable motor neurons. And again, doing patch clamp electrophysiology, showing very good basic function. But if you looked at it a little bit closer, you see that there is the membrane potential differences between the two phenotypes. So we know that there is an electrophysiological difference between the wild type and this mutation, okay, which now has a target for the ASL. You can actually see that is decreased here, and see the cell body area is slightly decreased as well. And then we compare that to the ALS motor neurons. What happens in ALS, it takes about 20 days to develop that phenotype of that deficit. With the CMT neurons, it took about 5 days. Okay, so we're seeing a difference between the ALS and the CMT, even though overall you'll see the functional deficits, but there are differences we can tell in these models between different disease states that have some of the same pathology shared. Okay, and actually we can then again put the systems in a neuromuscular junction chamber. It's basically monitor the neuromuscular junction over time. We see some interesting changes here. We don't see much fidelity, but again, CMT is not like ALS. You don't get spasticity. That's not part of the phenotype. where it is with ALS. We can see those differences in the model. So where we do expect to see a change is in the fatigue index, and we do that. You see a very quantifiable change, okay, between fatigue index. As we go through culture, we can monitor it from 5 days up to 16 days. You see here, basically, this is the tetanus is maintained in the wild type. What's really interesting is that we get a higher initial fatigue index, but then it drops off quickly. And we see, actually, we're looking at different variations of how the tetanus falls off in the system. It's very, very interesting. Okay, we've now got this to the point now, this is now being treated with ASO, and we're collecting that data right now to try to reverse it. What we're hoping to do is to go from patient cells to a evaluation of therapeutic in less than a year. Okay. We also have a model of myelination. We published this, ACS Chemical Neuroscience, in 2020. We showed very nice myelination efficiency. We've done this for primaries. We've also now recently done it with the induced pluripotent stem cells. Now with the induced pluripotent stem cells, we can add into our myelination model, okay, the disease phenotypes as well. But the company saw this. True North, who was acquired by BioVerda, who was acquired by Sanofi, basically saw this and said, well, okay, can you actually create a conduction velocity model for us? To be able to look at CIDP and MMN, which are two rare diseases based upon have a loss of conduction velocity in their systems. Okay. And we're able to, again, they were looking at complement deposition. Can you go back? Can I go back? Oh, well. Looking at complement deposition and basically showing their molecule could block complement deposition, but what they really wanted to do is look at conduction velocity. They basically took a modification of a neuromuscular junction chamber. What we did is you put electrodes in the tunnels to be able to grow the axons over the tunnels to be able to look at conduction velocity. Again, a direct clinically relevant functional readout, which is we're looking at conduction velocity of the axons over the microelectrodes. Okay. And again, what we're doing here, we wanted to see, we're only picking out conduction velocity over 3 electrodes, okay, to really show that we are actually getting that conduction velocity, we're not just getting electrical events. You can see here a range of frequencies. And again, what we did, this is the key experiment, is you can see here the untreated, the conduction velocities, the serum plus the isotype control of their molecule, serum plus their molecule. You see again, the conduction velocity maintained here, it was totally abolished, okay, with the isotype control, but restored with their molecule. And this was the only data that was utilized, okay, in an IND to enable this clinical trial. And this was published with Sanofi in Advanced Therapeutics. So we're getting regulatory aspects of this. And now we're actually then taking the motor neurons from this CMT2S, we're putting into this model here. So we have both neuromuscular junction formation and conduction velocity data that we can go into that IND that will go in for the clinical trial, one that we're doing. I just want to go very quickly show we can also put barrier tissues into these systems, BBB, GI tract. We can also look at kidney and skin. But we can actually now create a BBB GI tract CNS system. Again, getting very nice immunostaining, showing that we have the tight junctions of the models, and be able to show we can actually then get the GI inserts. We get, again, nice immunostaining, the BBB, expression of the transporters for glucose, PGP. And again, be able to show that we can actually get, this kind of a little bit washed out here, we can actually get transport through the system where we can actually then attenuate that on the BBB, but also attenuate it in the GI tract. So we can actually then model oral IV versus intrathecal dosing in a continuous system that then has again, the slide's a little bit washed out. On this side, can you see it on that side? Let's wash that on both sides. Basically, it shows we can actually put them together. We can also have systems where we can go and make multi-organ systems. This is what we're doing at looking at opioid overdose and recovery, and but also off-target toxicity. The nice thing about that is I wanted to make is we can actually create PK/PD relationships to these systems where we account for molecule absorption, metabolism in the system, be able to get the pharmacokinetics, and be able to get the pharmacodynamics for cardiac for inside and outside, inside of the cell and outside of the cell, get the pharmacodynamics. We use this data in collaboration with AstraZeneca, and be able to show a very quick correlation with taking PK/PD of our in vitro system and correlate that with in vivo data for guinea pig, dog, and non-human primate, This is published in Nature Scientific Reports. And then lastly, you also be able to create a recirculating immune system, innate immune system, okay, where we can actually take, showed in this paper in collaboration with Roche, that we can actually get the restorative macrophages and the inflammatory macrophages in the same system. So this is, I thank all the folks. This is a combination of the folks at UCF and Hesperos. And then ready for questions. Over here.
Yeah, can you comment, maybe I missed this, on the phenotype of your motor neurons? I mean, spasticity in theory should be coming from upper motor neurons, and the weakness should be coming from the lower motor neuron primarily. What are you modeling in your ALS chip exactly?
We're showing deficits at the neuromuscular junction, right? So that could arise for transport issues from the motor neuron to the muscular junction, which I think is part of the spasticity. Okay.
I don't think that's true. I mean, I'm a neurologist. Well, I don't know, but you're a neurologist. It's pretty much an upper motor neuron phenomenon. So I guess what I'm asking is whether you obviously are seeing an electrophysiologic deficit. I'm not arguing with that. What I'm questioning is whether you're really modeling spasticity.
Well,
your dropout. It's dropout, right? The dropout is your yeah.
Well, basically, you're missing contractions. So you stimulate a motor neuron, and you're missing contractions, which is probably due to some sort of transport. But the point is that we have modeled the fidelity of it and the fatigue index, but this has been accepted by the regulatory folks for a clinical trial. So it may not be exactly what you're expecting from a clinical standpoint, okay? But a lot of the aspects of what people believe clinically, okay, is sacrosanct is generally you have aspects of it in other parts of the system. So you might have a combination, okay, of effects between the motor neurons, but it doesn't make it invalid as a test, okay, for a disease model. Right? As he said, as Bob said, all models are wrong. Right? How useful are they? If I can get the regulatory folks to accept this, okay, for being able to get into a clinical trial, okay, why does it have to have a one-to-one correspondence to everything that you're going to see clinically?
Well, because you want your clinical trial to work, right?
Well, what the point is,
I mean, the better. I don't really understand your. I mean, you want the best model you can get.
right?
I don't think whether the FDA accepts something or not is really the gold standard for whether something is good. I mean, if you're running,
well, I don't know. I think a lot of people would disagree with that statement.
Other people are hiring you to get things through the FDA, then that's your gold standard. But from a scientific standpoint, I don't think that should be the gold standard.
We're working with 12 different pharma companies with these models, okay, on evaluating their therapeutics, okay, just in the NMJ model, okay. We have probably 3 or 4 active INDs that are going in with them, okay. From a purist standpoint, maybe you're correct, okay, but from a practical standpoint, you want models that work. Animal models, okay, have given really good insight to these diseases. But there's a reason why only 11% of drugs that use animal models ever get approved, okay? 11%, okay? So we have a different model here that may not be exactly what you want from a clinical purist standpoint, but it is giving very good, accurate data in terms of phenotype, evaluation of therapeutics. Okay, so I understand that this is totally new type of model. People haven't seen it before. You're going to get people jumping up and down and saying, well, it's not exactly what we expect to see clinically. But the animals, you're looking at 10% approval rates. So they're not really good models either. They're not really modeling what you see clinically for a human being.